Hello all. This video is going to explain the experiment on measurement of pH and conductivity. Measurements of pH and conductance find many applications in the fields of chemistry, biology, food technology, in monitoring of environmental pollution, industry, healthcare, etc. Therefore, this experiment has been designed to enable you to use a pH meter and a conductivity meter for determining the pH and conductance of any solution. Objectives. After performing this experiment, you should be able to operate a pH meter and a conductivity meter, calibrate a pH meter using a proper buffer solution, measure pH of a solution, determine the cell constant of a conductivity cell, and measure conductance of a solution. Let's discuss on measurement of pH. Measurements of pH and conductance is very important as discussed. We have studied the pH scale is a common convenient means of expressing the relative acidity or alkalinity of solutions. The pH of system plays a very vital role in success of many chemical and biochemical reactions. The pH of pure water is 7, therefore the pH of any water sample may be given an indication of the type of impurity present in water. In this experiment, we will measure the pH of the given solution. Requirements: pH digital meter 1 number, glass electrode, reference electrode assembly 1 number, beaker of 500 ml capacity 1 number, buffer solutions 2 numbers, distilled water 1000 ml, tissue paper a few. Principle and theory. Hydrogen ions in solution, like other ionic species, conduct an electric current. When a glass electrode is dipped in a solution containing hydrogen ions, a potential difference develops across a very thin glass membrane separating two solutions of different hydrogen ion concentrations, one within and the other outside the glass electrode. A pH meter is an electronic voltmeter that measures this difference of potential and through its internal calibration converts it to a pH reading which is displayed on a scale. The scale is normally taken extending from 0 to 14 pH units with the least count of 0.05 pH units or better. These days, digital pH meters are becoming more popular as compared to scale needle instruments. Let's now check out the procedure of conducting this experiment. Set the selector switch to 0 position and adjust the 0 position by a screwdriver if the pointer does not indicate 0. Mount the electrode assembly, a glass electrode and a saturated caramel electrode or a combination electrode in the clip on the stand. Wash the electrodes well with distilled water. Connect the power cable to a 220-volt AC supply. Switch on the instrument and wait for a few minutes till the instrument warms up. For calibration, take about 20 centimeter cube of standard buffer solution of pH 7 in a beaker. Lower the electrode assembly into it. It should be ascertained that the glass electrode membrane is completely immersed in solution. The electrodes should not touch each other or other sides of the bottom of the beaker. Swirl solution in the region of the glass electrode surface gently so as to bring it into pH equilibrium. Measure the temperature of the solution and adjust the temperature compensation knob to this temperature. Put the selector switch to a suitable pH range 0 to 7 for acidic or 7 to 14 for basic solution and adjust set buffer knob in a manner that the pointer reads the pH of the standard buffer solution placed in the beaker, that is pH 7 here. Put the selector switch back to the 0 position. Remove the electrodes from the buffer solution. Wash the electrodes with distilled water and wipe them dry with a tissue paper. Transfer the standard buffer to the storage bottle, as done in basic experiments in chemistry. You will need to make an additional adjustment when precise pH values are required in the acid or basic range. To do this, select pH 4 buffer for an acid. Repeat the calibration point again. Take about 20 centimeter cube of second buffer solution of pH 4 or 9.18 in another beaker and place the electrode assembly into it. Set the selector switch in a suitable pH range position and read the pH of the solution on the scale. If the meter reading does not agree exactly with the known pH of the buffer, set the slope control until the required reading is obtained. The instrument is now ready for measuring the pH of the unknown solution. Put the selector switch back to the zero position, remove the electrodes from the buffer solution, wash the electrodes with distilled water and wipe them gently with tissue paper. Transfer the buffer solution to the storage bottle. Now take about 20 centimeter cube of unknown solution in another beaker, introduce the electrodes into the solution, swirl the solution gently. Put the selector switch back in the suitable range position and read the pH of the solution on the scale. Put the selector switch back to zero position, remove the electrodes from the solution and keep the electrodes in water when not in use. Repeat the same for any number of samples taken. Now, let's perform this experiment on virtual lab. The first step before performing or testing the pH of a solution is to calibrate the pH meter. For this, take the buffer solution of 4 pH, click on standardization and check if the meter reads the pH 4. If the meter does not read pH 4, then adjust the knob in such a way that the display shows pH 4. Now take the second pH buffer solution, pH 7, click on standardization and check if the pH meter displays pH 7. If the pH 7 is not read in the pH meter, adjust the knob gently, such as the pH meter reads 7. Now calibration is done. Now select the water samples for which pH has to be determined. The first sample which we are going to consider here is tap water. Select the sample water, switch on the meter and check the reading. The pH of tap water is found to be pH 7. Switch off the meter, change the sample. The second sample is well water. Switch on the meter. The pH of well water is found to be 7.9. Switch off. Repeat the same for the remaining solutions. The third sample is the distilled water. Switching on the meter, the distilled water reads 5.8 pH. The fourth sample is sea water. Switch on the meter and the meter reads 7.8 pH. Now we have completed the calibration and testing pH of 4 samples that we have taken today. Precautions. Never touch the membrane of the glass electrode with anything else except soft tissue paper since it is fragile and is easily ruined if scratched or bumped. The electrodes must not be removed from the solution unless the selector switch is at zero. Never dip the glass electrode in a solution with a dehydrating action. If used for measuring pH of albuminous substances, the gas electrode must be cleaned with suitable solvents and then the electrode be placed in distilled water for a few hours before it is used to measure the pH of the other solution. For basic solutions with pH more than 1, glass electrodes of special compositions are required to avoid interference due to sodium ion. The glass electrode may be covered with a sleeve to save it from jerks. Let's discuss the design of a pH meter. pH is defined as a negative algorithm of the hydrogen. pH is defined as the negative logarithm of the hydrogen ion concentration of an aqueous solution. In simple terms, pH is a numerical value which quantifies the acidity or basicity of a solution. The number represents the number of hydrogen ions a particular substance released in the solution. On the pH scale, a pH of 7 is considered to be neutral solution. A solution with pH 0 to 7 is considered to be acidic, and above 7 till 14 is said to be a basic solution. In biological systems, pH is of utmost importance. Most biomolecules in our body function optimally because of a careful adjusted pH. Even in experimental systems, it is essential to maintain the desired pH to obtain accurate results. Hence, in biological experiments, pH is carefully monitored using equipments known as pH meter. The pH meter is a pH responsive electrode which measures the hydrogen ion activity in a solution and conveys the information to a voltmeter. The equipment consists of a two glass tube, each containing one electrode. The reference, the reference electrode is made of saturated KCl solution, whereas the sensor electrode contains a buffer solution of pH 7 and a silver wire coated with silver chloride is immersed in both these solutions. The sensor excuse me. The sensor electrode has a bulb at the end which is made of porous glass coated with silica metal salts. To measure the pH of a solution, this pH meter is dipped into the solution. Once the bulb of a central electrode touches the solution, the hydrogen ions in the solution replaces the metal ions on the bulb. This metal ion replacement causes a flow of current in the metal wires, which is read by the voltmeter. Let us look a little more closely at the scientific aspect of the pH meter. How does the pH meter convert the voltage difference into a pH reading? There is a simple equation which explains this, the Nernst equation. Through this equation, the pH meter converts the voltage difference into hydrogen ion concentration. The pH can be calculated using this equation. The pH meter is one of the most extensively used experiments in biological laboratories. To ensure accurate readings, the equipment must be regularly calibrated. Here is the Nernst equation. The pH meter detects the change in the potential and determines H plus concentration of the unknown by the Nernst equation. Here, E is the total potential difference measured in millivolts. ER is the reference potential. R is the gas constant. T is the temperature in Kelvin. N is number of electrons. And F is Faraday's constant. The pH of the samples that has taken has to be observed and observation. The sample solution that we have used in the calibration and observation. The pH of the sample solution taken has to be noted down, and using Nernst equation, the voltage difference has to be calculated and tabulated in this result. Thus, the pH of the sample solutions are noted and tabulated. The next part is measurement of conductivity. We have already studied the principles of conductometry, which deals with the measurement of electrical conductance of solutions containing ions. Measurement of conductance is potentially a very sensitive method for measuring ionic concentrations. It has many applications in chemical analysis. Measurements of conductance is one of the best ways for checking and monitoring the purity of water. A continuous recording of conductance is helpful in monitoring the purity of water purified by ion exchange or obtained from large distillation units. Conductometric measurements are also used to ascertain the endpoint in many titrations, example acid-base precipitation and complexometric titrations. There are also used in determining equilibrium constants, example ionization constant of weak electrolytes, solubility products of sparingly soluble salts, hydrolysis constants, etc. The objective of this experiment is the objective of this experiment is to find the conductance of the given samples. Requirements: conductivity meter 1 number, conductivity cell 1 number, beaker with 500 ml capacity 1 number, distilled water 1000 ml, tissue paper a few. Principle and theory. Solutions containing ions conduct electric current due to the movement of ion towards oppositively charged electrodes. The ability of a solution to conduct current is given by its conductivity. The conductivity of a solution is proportional to the number of ions, the charge on each ion, and the speed with which the ions move. The speed of an ion in turn depends on the nature of the ion, nature of the solvent, presence of other ions, temperature, and the applied field. The conductance G is defined as the reciprocal of resistance R. That is, resistance is measured in ohm, conductance measured in Siemens. The resistance of a homogeneous body of uniform cross-section is proportional to its length and inversely proportional to the area of its cross-section A. The proportionality constant P is called this resistivity. The unit of P are m. The conductivity K is the inverse of the resistivity. The units of K are Sm power minus 1. Procedure. Plug in the instrument to the power supply. Put the frequency selector switch to the required frequency, say 1000 Hz. Keep the sensitivity knob roughly midway between the lowest and highest sensitivity. Clean the conductivity cell with distilled water. Connect the conductivity cell electrodes to the appropriate terminals of the instrument. Take the standard KCl solution in a clean beaker. Introduce the stirring rod in the solution and put the solution beaker on a magnetic stirrer plate. Insert the conductivity cell in the solution. Ensure that the platinum plate electrodes are completely immersed in the solution and they do not touch the stirring rod. Switch on the instrument and allow it to warm up for 2 to 5 minutes. Measure the conductance. For first adjust the course balance by rotating the change. First, adjust the course balance by rotating the range selection knob and then increase the sensitivity to obtain the fine balance. Convert the reading for the proper value by multiplying the factor and used on the range selection knob. Remove the solution from the beaker and wash conductivity cell properly with distilled water. Take the unknown solution in the beaker and measure its conductance in the manner as for standard KCl solution. Calculate the cell constant from the conductance and conductivity values of the standard KCl. Calculate the conductivity of the unknown solution. For titration work, the value of cell constant is not required to be calculated since the cell constant will remain unchanged during the course of given titration. Precautions. The conductivity cell, when not in use, should be kept in distilled water to prevent drying of the platinum electrodes. In case of fouling of the conductivity cell electrode plates, clean them by keeping in dilute potassium chromide solution containing H2SO4 solution, dilute chromic acid for 24 hours and then washing with running water followed by the distilled water. Let's perform the experiment. Use KCl solution for standardization. Click on standardization and check the electrical conductivity of the given KCl solution. It is found to be 1412 microsiemens per centimeter. Now select the samples totally pure water, switch on the meter and check the result. The meter displays 0.055 microsiemens per centimeter. Now switch off the meter, change the sample solution. Distilled water is taken now. Switch on the meter. The electrical conductance of water sample is 0.5 meter. The electrical conductivity of distilled water is found to be 0.5 microsiemens per centimeter. A third sample solution is taken, RO water. Now switch on the meter. Electrical conductivity is found to be 68 microsiemens per centimeter. Repeat the same procedure for n number of samples taken. Observation. For calculating cell constant and conductivity, the given formulas has to be applied. Result. Thus, the conductivity of the sample solutions are noted and tabulated. Thank you.